and the basic cell physiological processes show a slight change, and these changes focus on both of photosynthesis and respiration. At least we concluded from the attenuation of the CO2 uptake and the stimulation of CO2 production. And we also saw a triggering of hyperpolarization, presumably due to the activation of the proton ATPase. Important is, or one of the most important points, is that all the observed reactions are of transient nature. They are present in the cells in the first half an hour or first couple of minutes, and after that the cells gradually get accustomed to the new conditions. So the conclusions which we can draw is that aquatic plants are suitable experimental objects to study deuterium depletion, because they show a rapid and relatively significant change in their physiological pattern. And we think that this change in the cell physiology might be attributed to the change in the hydrogen over deuterium ratio, but we also saw that the plants quickly adapt to the new conditions, but these early actions can be exploited to study further what deuterium depletion might be doing in cells. Thank you for your attention.
The talk is open for questions. Go ahead. Can you wait for the microphone? Just 1 second.
Have you tried to estimate for the overall effects of the immersion in DDW of the plants? How long did you keep them?
No, that's the point, and that's why I asked you in the break that how long time did you need when you observed the increase in the growth rate, because we concentrated only this first hours, and frankly speaking, we didn't see any longer actions, and you gave me a very good indication that we might do that.
And are there any possibilities of chemically blocking the hydrogen transport through the cell membrane, the vegetal cell membrane? Because I don't know.
There are compounds which can be used in isolated plasma membrane vesicles. For example, vanadate, sodium orthovanadate is one of them. But I'm not aware of any studies which show that they work in vivo as well. But maybe it's a textbook story. We have to find it out.
I'd like to ask you a question. Do you think the fast adaptation, because this is how I would call it, to deuterium depletion in the plant system is consistent with the fact that plant enzymes, for some reason, especially the ones that are involved in carbon fixation and eventually photosynthesis, are just more discriminative of isotopes? Meaning that, because we do know that although there is no isotope discriminating effect in mammalian systems as far as intermediary metabolism go, if we apply the same concentrations of stable glucose, stable isotope labeled glucose, in plant systems there is, in fact, discrimination against the heavy glucose isotope. Do you have any idea or any answer why this may happen, and why there's different enzyme responses in plant versus mammalian systems?
Well, I don't know if if we can say anything at the enzymatic level. But what we think is there might be a balance between photosynthesis and respiration in plant cells, because environmental factors usually affect them anti-parallel way. And it's tempting to speculate that this switch, which can find a balance between the two processes, might be the one which is sensitive to hydrogen and deuterium. But the exact nature of this mechanism is not known.
Last night, as I was just finishing my conclusions, I was really almost tempted to go into this metabolic contra-analysis field, which I'm not the expert in. We do have a collaborator from the University of Barcelona who is really the main guru in the field. And I was reading the book chapter we wrote together. Obviously, she wrote the part which relates to this enzyme elasticity, meaning that there are an elastic component of the enzyme function, meaning that in certain environmental factors, and probably deuterium depletion is one of the factors that can affect the kinetics of enzyme reactions just based on whatever else is needed, for example, a hydrogen for reducing, yet it's not really the main function of the enzyme to separate hydrogen in water, but actually uses that hydrogen molecule to, for example, synthesize fatty acids de novo. And even though there's a main regulation from the substrate point of view, as far as what the substrate availability and what the product level, positive, negative feedbacks on enzyme allosteric regulation, it seems that actually deuterium or hydrogen, which is practically just the other reactant in the reaction, but not the carbon source, can dynamically change the kinetic patterns. And this was just one area that I have been I was not ignoring, but I'm really not truly the expert, but we have collaborators who allow us just to go and get or gain more insight into these fields. And I'm just talking because we have a few minutes, so and I think it's really interesting. But based on your studies, it seems to me that actually plant enzymes have even more of an effect of this sort.
Yeah, that reminds me that in the first talk, Gabor mentioned that there are differences in plants carrying out different pathways of photosynthesis. For example, the C4, C3, and CAM pathway respond differently. Yeah, is this correct? Yeah.
Regarding that, so the C, if it depending on what type of food we eat, for example, pea or popcorn, it could be more depleted or less depleted for deuterium, the carbohydrate, and the CAM plants even can enrich the deuterium, so depending on which metabolic pathway is present in the plant. So it can discriminate. The RJ can discriminate in light between hydrogen and deuterium, but not in dark. So this is what I say, that it is present, I think, in the living organism.
Actually, this is how we know where the marijuana is coming into Los Angeles, because if it's from Colombia, it has a different deuterium and carbon enrichment than if it's from Mexico. Yeah. I mean, it was grown in Mexico. Yeah. We know because we work with reference laboratories, just for that particular purpose. So it's very clear. Yeah, it's very evident.
Yeah. Yeah. Okay.
Thank you.
Thank you very much. So.